Hi there, I'm Mohammed Kudus Rao and I'm the founder of BioGen Therapeutics. So I'm building BioG-X1.0. That is basically an AI-powered CRISPR decision intelligence system. So my focus is simple: to help turn fragmented gene editing workflows into structured, explainable decisions. That is basically an early stage AI project. So it is still being built with a serious long-term vision. Let me start with the problem that I'm trying to solve. So, CRISPR design is powerful, but the workflow is still fragmented. Target identification, guideline design, of target assessment, and efficacy scoring are often handled as separate steps. So, that matters because each step is isolated. You lose the bigger picture, you are no longer making integrated decisions across safety, efficacy, and target selection. So, and in therapeutic design integration, exactly what you need. So that leads to the core insight behind the BioGX 1.0. The key insight is that these decisions are not independent, they are interdependent. Target selection affects guide performance, guide design influences off-target risk, and optimizing one parameter in isolation can reduce overall therapeutic viability. The real challenge is not just generating these options. The real challenge is prioritizing the right op based on how the variables interact together. So that is exactly why I designed the system the way I did. The BioGX 1.0 is built as an AI-assisted CRISPR decision framework. So let's start with gene and disease input, then the strategy engine generates candidate editing approaches. The scoring layer evaluates efficacy and off-target risk, and the explanation layer provides the reasoning behind the final recommendations. So the output is not just a result, it is ranked explainable decision. That structure matters because I'm not trying to build another isolated tool. I'm trying to build a framework that integrates the decision process from start to finish. And at this stage, the project is already real. At this point, right now, BioGX is already in active development. So I built a modular prototype with independent testable components. There is a streamlit-based interactive interface, and a core logic is separated into strategy, scoring, and explanation modules. That modularity is intentional. It gives the system a clean foundation, and it makes the architecture easier to expand, validate, and improve over time. So let me show you, what it looks like in practice. This slide shows the two sides of systems. So on one side, you see the modular Python code base. On the other side, you can see the working prototype interface. So that combination is important because it shows that it's not just a concept or a slide deck. It is a functioning system with a real structure behind it. And as a developer, that's a standard I want to keep in the future. The real implementation, not just presentation. From here, the next step is to make the platform broader and future ready. The long-term vision is to make BioG-X 1.0 editor agnostic. That's the future goal. That means the system should be able to work across multiple CRISPR data types, including Cas9, prime editing, base editing. So I want the architecture to stay modular, extensible, and research-grade, so it can evolve. So it can evolve as the field evolves. And in the future, I see that becoming a system that can continuously learn from experimental and literature data. So to reach that vision, that development has to move in clear stages. The roadmap is structured step by step. First the prototype stage where the core modules are implemented and tested, and then the MVP stage where the workflow becomes more integrated and interface is refined. After that comes data integration, research validation, and then platform expansion. That sequence matters because it keeps the project disciplined. It moves from prototype to validation in a way that is technically honest and scientifically scalable. That brings me to closing point. To conclude, this BioGX 1.0 is being built as a structured long-term platform. So it is rigorous, modular, and redesigned and to scale with the science. So right now it is active development and moving from prototype towards our research-grade system. So that is the direction I'm committed to build by step by step. So I hope there will be more of this in future. So stay updated. So for the latest updates, you can visit the website of our official company I've launched, and you can also check the updates on LinkedIn profile. We're looking forward towards more collaboration with scientific institutions and other scientists, and we're also looking for early stage investments if you're interested. So have a nice day.